spike protein on the coronavirus, it finds a surface protein on one of your cells, this big blue blob that is one of your human cells. And on this human cell, there's a surface protein. It's a receptor called the ACE2 protein. The ACE2 protein is found pretty commonly on lung cells and on the cells lining the airways. And so it makes sense that this particular virus, one of the symptoms of the disease it causes is respiratory distress, because it specifically targets cells that have this ACE2 protein on them, which is primarily your lung cells. The spike protein, it binds to the ACE2 protein. And when it binds to the ACE2 protein, it sends a signal to the cell to bring the virus inside of it. And once it brings the virus inside of it, the virus opens up and releases that genetic material. So the genetic material, some of it goes on to get copied over and over and over again by your cell's machinery. But some of it goes down into a part of your cell called the endoplasmic reticulum, or the ER for short. In the ER, particularly in the rough ER, there are ribosomes. The ribosome is a protein complex that has the job of reading the messenger RNA instructions and turning them into each of the protein components of the virus. The messenger RNA goes to the ER, the endoplasmic reticulum, and the endoplasmic reticulum gets to work on making the spike protein, the membrane protein, the viral envelope, etc. And then once you have all of the pieces of the virus, the next step is to assemble them. And that assembly happens in what's called the Golgi body. The Golgi body assembles the viral proteins together along with some of the messenger RNA. And once it's all assembled, the complete virus is sent out of the cell via a process called exocytosis, and the virus goes on to infect another cell. But this messenger RNA that's left behind, it gets copied over and over and over again. So it gets copied, sent down to the ER. The ER makes the spike protein, the membrane proteins, the viral envelope, and it all gets packaged together in the Golgi body, and then it makes a new virus and it sends it out. Your cell has effectively been hijacked into making virus. As the cell is releasing the virus, you have killer T cells who have been trained to spot maybe the spike protein or maybe the membrane protein. So they see this cell releasing virus. Killer T cells will find this cell and they will kill it. Now remember, this is one of your cells that's involved in your airways. This is one of your lung cells. It could be lining the alveoli, which is where the oxygen exchange takes place between the blood and the lungs. So if your killer T cells are destroying your lung cells because they've been hijacked, you're going to suffer from difficulty breathing. That's one of the reasons why respiratory distress is one of the biggest symptoms of COVID-19 infection. Now that we know how our enemy works, we're going to talk about the types of vaccines we have available in our arsenal. The first is called live attenuated. This is where you get a small amount or a weakened version of the live virus. Now, the good news with these live attenuated vaccines is that they provide a very strong response by the immune system, and so you have long-lasting immunity to the virus. The downside is you are getting whole virus. These vaccines are risky because, in theory, if your immune system isn't able to shut down the virus before it has a chance to replicate in your body, then you could develop the full-blown infection that is caused by this virus. This was not going to be the type of vaccine that was going to work for this massive vaccine campaign. The other thing that we have is an inactivated vaccine, which is where you get the whole virus, but it's been deactivated or, quote, killed. Killed is in quotes here because viruses are not technically alive. They lack a metabolism and they lack any machinery for their own replication. They rely on your cell's machinery to do it. But we can deactivate them. The downside to these vaccines, while they are safer, is that you might need to get several booster shots with this type of vaccine in order to maintain that immunity. Next, we have subunit vaccines, which is instead of getting the whole virus or a small amount of the virus, you're getting a piece of the virus. And what's great about these types of vaccines is that there is zero chance that you are going to become contagious. The downside to these vaccines are that you have to make that piece of the virus in a factory in these big tanks in these cells. And so it can take a long time, and it can be very difficult to do, and it can be very expensive. But we also have another type of vaccine called mRNA vaccines. These are relatively new vaccines where instead of making the piece of the virus, instead of making the subunit in these big factories in these tanks of cells, the mRNA vaccines give the patient the instructions to make the spike. Instead of it getting made in a factory, it gets made in your body. But just the spike, the protein that binds to the ACE2 protein on your cell to get inside of your cell and hijack it. Active ingredient in the Pfizer-BioNTech vaccine is messenger RNA. Messenger RNA is genetic material that will, in this case, encode the viral spike protein of the coronavirus. Messenger RNA, it only codes for the spike, not the membrane proteins, not the viral envelope. Why would they bother to put anything else in this vaccine? They needed to in order for this vaccine to actually work. So messenger RNA is the key ingredient. It's the active ingredient in this vaccine. But messenger RNA is something that we call hydrophilic. It loves water. It loves being surrounded by water. Now your cells, they are surrounded by what's called a plasma membrane. It's the skin of the cells, and it's very oily. And so we call it hydrophobic because it doesn't love water. It's water fearing. Now, messenger RNA, being something that loves water, can't get across the oily membrane of your cells that hate water and don't want to be surrounded by water. So the question is, how do you get something that loves water across a membrane that hates water? In order to get the messenger RNA inside of the cell, we're going to use lipid nanoparticles. And lipid nanoparticles are made up of lipids. Lipids are things like fats and oils that are naturally produced by our body. They have these really long names, but that's just so that chemists can be very precise when describing these different lipids. So the way that this works is you've got a sphere of oil and you open it up, and inside you put the messenger RNA. Then this lipid nanoparticle is going to merge with the cellular membrane. And when it merges with the cellular membrane, it's going to release the messenger RNA inside of the cell. So the purpose of the lipid nanoparticle is to transport the messenger RNA across the plasma membrane of the cells. Next, we have pH buffers and salts. pH refers to how acidic or how basic a solution is. So if you were to get hit in the face with a very strong acid or a very strong base, your face is going to melt off essentially, or it's going to burn. And so lipids are very susceptible to changes in pH and changes to acidity or how basic a solution is. And so we put these buffers in there that will prevent the solution from becoming more acidic or more basic than we want it to be. Next, there are salts, which also aid in making the vaccine more comfortable to administer. Last, we've got a stabilizer, which is sucrose, and this is used for when the vaccine is repeatedly frozen and thawed. The sucrose helps to stabilize it during that transition. So a couple of things. The first is that it doesn't contain any mercury, thimerosal, or preservatives. The reason why this creator decided to include this in their slide is because there is a debate on the internet, because the internet is rampant with misinformation. There is a debate that vaccines cause autism because vaccines contain mercury, thimerosal, which is some very high percentage made out of mercury, or because these vaccines have preservatives in them. And regardless of where you stand on that particular issue, this vaccine doesn't contain any of that. So the discussion is moot there. There is nothing in here that has ever been argued to cause autism. Further, vaccines do not cause autism. but that's a debate for a different day. Vaccines do not cause autism. This vaccine doesn't contain any of those ingredients. It didn't need any of these. Now that we know what's in the vaccine, we're going to talk about the Pfizer-BioNTech safety and efficacy. So we're going to go over a couple of different things. First, we're going to talk about the safety. So to understand the safety, you have to understand what sorts of things they were looking for in these vaccine trials. First of all, there were over 37,000 participants in this trial. And the way that vaccine trials work is they take half of the participants, and they give them the actual messenger RNA vaccine. That's going to be this column on the left. The other half, they give something called a placebo. The placebo is essentially a shot that has nothing in it. The placebo is given to people because of a couple of reasons. The first is we want to know what percentage of people can you expect to experience certain symptoms, whether they got the vaccine or not. For example, let's suppose that you get a vaccine and then you go and eat at a restaurant, and then later that night, you're feeling some sort of gastrointestinal condition. Maybe you have heartburn, or maybe you are vomiting. You don't know if it was from the vaccine or if it was from the restaurant.